All right, our next speaker is Nathan Yee from Rutgers University. He is a co-leader of Theme 3. Nathan got his undergraduate degree. He grew up in Canada, in Calgary. He went to McGill University for his undergraduate degree. And then he, I think, went to University of Leeds. Notre Dame. And then you went to Leeds for a postdoc. And he has been at Rutgers for, I don't know, 19 years, getting up there. So anyway, it's Theme 3 time. Now we bring in some minerals.
Thank you, Paul. Okay, so today I'm going to talk about the Rutgers half of Theme 3. So I'll present work from Jiwa Hao, Winnie Liu. Jiwa is now a faculty member at the University of Science and Technology of China. Winnie Liu was a PhD student in the Geological Sciences graduate program. She's now a postdoc at PNNL. I'll show work from Jennifer Goff, who's now a postdoc at the University of Georgia, and Orion Farr, who's a master's student and undergraduate here at Rutgers, and is currently doing a PhD in France. We'll hear a lot more from the other side of Theme 3, the Carnegie Group, from Shauna, Anirudh, Michael Wong, and from Dan Hummer. So I've broken up this presentation to 3 parts. The first part, I'm going to talk about our recent work on the geochemical cycling of sulfur. I'm going to just spend 5 minutes to recap a paper that was just recently accepted in Science Advances. Then in the second portion of the talk, I'll talk about our work on the geochemical cycling of phosphorus. And in the last part, I'll give you a quick update on the astrobiology course here at Rutgers. Okay, so we all know that sulfur is an essential element of living things. It's found in the amino acids cysteine and methionine. In the case of cysteine, sulfur plays numerous critical biological functions, including the maintenance of redox homeostasis, formation of disulfide bonds and cross-linking in protein structures, in metal binding, as well as electron transfer reactions in metabolism. So cysteine and methionine are synthesized inside the cell via assimilatory sulfate reduction. So sulfate is a soluble oxyanion that exists in the environment that needs to be transported inside the cell, where it's activated to adenosine phosphosulfate, then converted to phosphoadenosine phosphosulfate, and then it's reduced to hydrogen sulfide via the formation of sulfite. This hydrogen sulfide is then incorporated into cysteine via the action of cysteine synthase. So all 3 domains of life have evolved biological pathways to assimilate sulfate in the environment for the biosynthesis of cysteine and methionine. So the research that I'm interested in is, what are the geochemical processes that formed sulfate in the early oceans at the nascent stages of microbial evolution? So the main insight that we've made is that the early sulfur cycle on Earth was linked to the iron cycle. Okay. So 2 things you need to remember about the Archean world where microbes evolved is that 1, the atmosphere was anoxic, and because there was no oxygen in the atmosphere, there was no ozone in the stratosphere. Because there was no ozone layer, ultraviolet light could penetrate through the atmosphere and interact with superficial environments. The second thing you need to know about the Archean world is that the waters on Earth were ferruginous. That means they had high concentrations of dissolved ferrous iron. And when you have those 2 conditions occurring together, we had UV light that interacted with dissolved ferrous iron in the surface waters of the ocean. What happens is that the UV light can photo-oxidize this ferrous iron to produce ferric iron. Electrons are transferred from Fe2 plus to protons to generate hydrogen gas. This hydrogen gas can then volatilize in the atmosphere, and this ferric iron participates in a wide range of geochemical reactions. One of the reactions that we propose that can occur is that this ferric iron can chemically oxidize pyrite in sediments. Okay, so in the modern geochemical cycling of sulfur, all the sulfur in your body, in your cells, originally came from a rock. Okay, and it occurs through the chemical breakdown of pyrite. Okay, so we're trying to understand what were the geochemical reactions that broke down pyrite in the early stages of microbial evolution when there was no oxygen in the atmosphere. So we're proposing that ferric iron could oxidize pyritic sulfur to form sulfate and release ferrous iron, and this reaction produces lots of acid. So I drew a picture to kind of illustrate this geochemical process. So this is what I'm calling the photochemical weathering hypothesis, where ultraviolet light photooxides ferrous iron to ferric iron. This ferric iron attacks pyrite to generate sulfate that can be assimilated to the biosphere and release ferrous iron. This ferrous iron can then be reoxidized by light to continuously sustain this process. Okay, to test this hypothesis, we constructed this experimental setup where there's a UV lamp in the center that's surrounded by 6 UV transparent quartz reaction vessels. We can place known amounts of crushed mineral powder, in this case pyrite, suspended in deoxygenated water, crimp and seal it, and do all these experiments under strict anoxic conditions. So in this experiment, we added some dissolved ferrous iron and mixed it with pyrite grains. And when we turned on the lamp, and then we sampled the headspace and the aqueous suspension to track the geochemical reaction. Okay, so this is what we observed. When you turn on the light, the gas composition in the headspace changes dramatically. Okay, and we see an enormous amount of hydrogen gas being produced. Dark controls, we see no reaction. So this is a plot of hydrogen concentration over time. And what we're essentially seeing is the photooxidation of ferrous iron to produce this hydrogen gas. By the end of the experiment, we had upwards of over 2% of the headspace was hydrogen gas. Okay, so that's a lot of hydrogen. Then we analyzed the aqueous solution to determine the amount of sulfate that was produced and track the changes in pH. In the dark control experiments, so these are the quartz cuvettes wrapped in aluminum foil, there was no reaction. There was no change in sulfate concentration, no change in pH. When we turned on the light, we saw a lot of sulfate being formed, millimolar concentrations of sulfate being formed, concurrent to a decrease in pH. So what's going on is the ferrous iron is being photooxidized to ferric iron, and the ferric iron in turn chemically oxidizes the pyrite to form sulfate. Okay, we also tracked the release of transition metals from the pyrite structure. Bob Hazen tells me there's no such thing as pure pyrite. All pyrite contains transition metals in their mineral structure, including copper. So in the dark, there's no copper release, but when we chemically oxidize the pyrite and mediate the photooxidation of the pyrite sulfur, we see copper release. So just to clarify Jana's point, in the primordial soup that you envision, in the Archean Earth, where it was a water world, there was still very, very little copper in seawater. There was very little. It wasn't until you started forming land and continental crust that you started seeing chemical weathering of pyrite and the delivery of copper into the oceans. So the take-home message here is that sunlight can chemically break down rocks, and the amount of sulfate in the ocean was dependent on how much land was exposed to sunlight. Okay, in the second part of the talk, I'm going to talk about the biology of phosphorus. As you all know, Phosphorus is found in numerous biological molecules. It's a critical component of nucleic acids, phospholipids that make up membranes, ATP, phosphates involved in cell signaling, as well as post-translational phosphorylation. And after the rise of animals, phosphate minerals became critical components of our bones and our teeth. So similar to the sulfur cycle, phosphorus in the ocean comes from the chemical weathering of rocks on the continents. So the breakdown of rocks on the continents delivers phosphorus through streams and rivers. Once it gets into the ocean, it's assimilated by marine biota. The key thing to know about the marine phosphorus cycle is that phosphorus is quickly and rapidly recycled from detrital organic matter. So life has evolved enzymatic pathways, for example, alkaline phosphatase that will cleave off the phosphate functional group from organic molecules and quickly assimilate that inorganic phosphate and reutilize it in the ocean. So we were curious whether or not there might have been photochemical mechanisms that recycled phosphorus before the evolution of alkaline phosphatase and enzymatic pathways. So our hypothesis is that ultraviolet light can either directly or indirectly break apart organic phosphorus molecules to release inorganic phosphate in aqueous solution. The three compounds we examined in this study were adenosine monophosphate, phosphotidylserine, and methylphosphonate. Okay. So in this plot is the amount of phosphate released from these molecules when irradiated by ultraviolet light. The red is AMP, the green is phosphotidylserine, the blue is methylphosphonate. For all three molecules, UV light broke these molecules apart and released the phosphorus in the form of inorganic phosphate. Okay. Reactions with AMP and phosphotidylserine were very rapid. Methylphosphonate, shown here in blue, slightly slower. There were no reaction found in our dark controls. So what this tells us was that UV light was involved in phosphorus recycling in the Archean oceans. These observations also raise interesting questions about the habitability of the photic zone during the early evolution of life, because they would have to overcome these chemical reactions mediated by light, which broke apart these important biological molecules. So this paper we've submitted, have reviews back. We're in the process of revising this manuscript and resubmitting it hopefully in the next week or two. The last paper I'm going to talk about is Jiwa's paper on the Enceladus oceans. Okay, so Enceladus is a moon of Saturn. It's a very interesting moon that has a crust of ice and a subsurface ocean. In the south pole of this moon, we observe geysers emitting large amounts of water, and this was observed through the Cassini mission. Based on density measurements, we know that this moon has a rocky core that's likely heated through tidal heating through its interactions with Saturn. We think there are hydrothermal vents in this subsurface ocean. So this is a fascinating astrobiology target. What Jiwa asked was, if this rocky silicate core had a chondritic composition, how much phosphate would be dissolved in the Enceladus oceans? First thing Jiwa did was try to determine the chemical forms of phosphorus that might exist in the Enceladus ocean. It turns out that the chemical speciation of phosphorus is dependent on pH and the redox potential, shown here in terms of the log hydrogen fugacity. Okay, so this is basically an EH-pH plot, where high hydrogen fugacity means you have reducing conditions. Low hydrogen fugacity means you have oxidizing conditions. Based on the observations of Cassini, we know how much hydrogen gas is in the waters of the Enceladus ocean. And even if we take into account seafloor pressure, what GWA discovered was that the chemically stable form of phosphorus must be in the form of phosphate, or phosphorus in the +5 oxidation state. You need much more reducing conditions to get phosphite, and you need much more reducing conditions and as well as acidic waters before you get phosphate. Okay, so phosphate is predicted to be the chemically stable form of phosphorus in these waters. The next question that GWA tried to answer was, well, how much phosphate can we expect to be in these waters? And he found that there were 3 master variables that determine how much phosphate can dissolve in these waters. One is pH. pH determines solubility of phosphorus-containing minerals. The master principle here is how much phosphate can be in these waters. Well, the concentration of dissolved phosphorus is controlled by the solubility of the least soluble phosphorus-bearing mineral phase. Okay, so that's the core principle. The solubility of the least soluble phosphorate mineral is dependent on pH. And there are 2 other variables that he found were very important. One is carbon dioxide, and the other is the ammonium concentration. Now, high carbon dioxide, dissolved carbon dioxide in water actually promotes phosphate mineral dissolution. That's because dissolved carbonate binds with divalent cations that forces the phosphate minerals to dissolve more, to re-equilibrate with that water. Ammonium has a different effect. Ammonium can actually bind with dissolved phosphorus to precipitate ammonium phosphate minerals, for example, struvite. So high ammonium promotes phosphate mineral precipitation. And based on these constraints, he's estimated the concentrations of phosphorus that might exist in the Enceladus Ocean, shown here in this shaded gray. This paper, Bob Hazen and I are co-authors, as well as several others on this paper. It's been submitted to PNAS. We've had the reviews back. GWA's made the revision, and I think he just resubmitted the revised version to PNAS yesterday. Okay. All right. The last part of my talk, I'll talk about this astrobiology course that we've now offered for the 3rd time here at Rutgers. It fulfills our School of Arts and Sciences core curriculum goals. So we get a lot of students from non-science majors enrolling in this course. So there are history majors, communication majors, philosophy majors even taking this course. Just give you a quick snapshot of what I cover in this course. Because the audience is a non-science major, I start with very basic concepts, starting with the periodic table, talk about molecules, including nucleic acids. Then I go to some higher level concepts. We talk about DNA replication, transcription, translation. And by this time I'm forcing them to learn a lot of biology because I'm a strong believer that in order to understand the origin of life, you need to know something about what is life. We talk about microbiology, both bacteria and archaea and eukaryotes, microbial ecology, early habitats for microbial life, extremophiles. I teach them how to read a phylogenetic tree, talk about the last universal common ancestor, mutation, evolution, endosymbiosis and the evolution of eukaryotes, history of life on Earth, the rise of multicellularity, the fossil record of life on Earth, meteorite impacts events and mass extinction, talk about the number of marine taxa over geologic time and the 5 mass extinction events that have occurred. We talk about the evolution of human species over time. We have a unit on geology. We talk about banded iron formations, the rise of atmospheric oxygen, plate tectonics, the carbon cycle, carbon burial, sediment diagenesis, formation of lipid biomarkers, the geologic record, the application of carbon isotopes to fingerprint metabolism. Then we do a planetary science section where we talk about the Viking lander experiments on Mars, current missions to Mars, Mars geology. the thermodynamic stability of water on the surface of Mars. Talk about this amazing moon, Enceladus, another amazing moon, this Europa, moon of Jupiter, and its subsurface ocean. The methane lakes on Titan, the largest moon of Saturn. Talk about star formation of the solar disk, and the distribution of chemical elements in the disk to form planets. Talk about how we detect exoplanets and distant stars. Talk about G-type stars, K-type stars, and M-type stars. Detection of intelligent life in the Milky Way galaxy through the Drake equation, and distant galaxies in the visible universe. The reason I'm showing you this course, B asked me to give you a synopsis, but really I'm asking for anybody that's interested in giving a guest lecture to please contact me if you want to contribute and teach in this course. We can do remote, in person. You tell me what you want to do, and I invite you to lecture in this course. I'll take any questions. Right, so the question is, okay, sure. So it's about phosphorus flux. How much phosphorus flux occurred in the Archean oceans based on photochemistry. I kind of think of flux slightly differently. One is how much mass was transferred from the continents to the ocean. That's kind of my first step, is how much phosphorus is entering the ocean. What I think about is the turnover time once it's in the ocean, is how fast it can be recycled, right? So that's the kinetics of photodegradation. Depends on the type of molecule, is how fast it gets recycled. But these rates are faster than enzymatic rates. Based on the photon flux of the early ocean, we have equal or faster rates than enzymes turning over these organic phosphorus molecules. Yeah, I don't think the photic zone was habitable at this time. There's another thing, which is if ATP breaks apart by light in minutes, really hard for life to overcome that. Maybe there's some pigments that dealt with it. So the scenario I'm really thinking about is the upwelling of waters from deeper regions and getting broken up in the photic zone to release this inorganic phosphate.
He is wondering whether you have any thoughts on the sulfate minerals that were most likely to form as a consequence of pyrite oxidation.
So if I understand the question correctly, is what type of phosphate minerals might have formed from this anoxic pyrite weathering reaction that we're talking about? So that's very interesting because, as Bob knows, the record of sulfate mineralization in the Archean is very sparse. I think there's only a couple of deposits at this period of Earth history that have preserved sulfate minerals, and it's almost always in the form of barite. That's simply because there was just very little sulfate in early environments, and you had to have these very unique restricted basins to get these evaporating deposits to preserve sulfate in water. So Bob probably knows this better than me, but I think barite's the one you have to look for.
I think Michael Wong has his hands up.
Hi, yeah, really good talk. This is Mike Wong from Carnegie. I'm really interested in the Enceladus work. That's a really cool study. To my knowledge, phosphorus-bearing compounds weren't detected by the Cassini mission. So I was wondering how your expectance of a high abundance of phosphorus jives with that, whatever upper limit could have been gained from the Cassini mission, if they're consistent with that. Be curious on your thoughts.
Yeah, so Michael's absolutely right. These are theoretical predictions, not direct observations or measurements. So the idea is, if we had a silicate core, if the silicate core has a similar composition as chondritic meteorites, could we leach phosphorus from those rocks into the aqueous phase? And what would be some geochemical controls on how much phosphate would be dissolved in those waters? So based on the solubility of those minerals that we find in chondritic meteorites, we think we could get enough dissolved phosphate for microbial life in those waters. I'll be happy to talk to you more about this work. It's lunchtime here and I'm 5 minutes over. So Michael, I'll talk to you soon.
All right, sounds good. Thank you.